So my name is Rui Lopes. I came from the UCL London School of Pharmacy. I just entered the project like 8 months ago, so probably I'm new for all of you. So how did they get to London? So as you can see, in the last years I've been running a marathon on nanomedicine. So I started early with liposomes, conventional liposomes for parenteral administration, followed by a PhD where I introduced solid lipid nanoparticles in addition to the liposomes in Lisbon. There's a familiar face, Manuela Gaspar. Then I followed to Coimbra, continue a little bit with the liposomes, with peptide target liposomes for breast cancer with Professor João Nuno, also a familiar face, lots of familiar faces here. And what was the next step? Continue with lipid-based carriers? Yes, it's a possibility, but science is about challenge. So I took the opportunity to join Professor Andreas Schatzlein group in the UCL School of Pharmacy, where I work with polymer-based systems, oral delivery, and hydrophilic compounds. So basically, I changed from lipid to polymer, parenteral to oral, hydrophobic to hydrophilic, so a big challenge. As I told you, I just joined the project like 8 months ago. So my tasks in this project were basically nanocarrier design, development, and characterization, and formulation of the peptides that the consortium selected. This seems relatively simple. However, as most of you know that work with oral delivery, having an objective that combines oral, peptide, and delivery is a challenge. It's not an easy task. In addition, as I told you before, I just joined the project 8 months ago, in the period that deadlines were urgent. Prototypes has to be sent to all the partners to be tested, so it was complicated, but it was possible. In London group, we work with GCPQ, just for short, quaternary ammonium palmitoate glycol chitosan, so it's a derivative of chitosan. We can adjust the properties of the polymer by adding palmitic acid, make it more hydrophobically, and quaternary ammonium groups. We can change the percentage of the modifications in both parameters, making different polymers depending on the needs that you want. It's very versatile. We can conjugate the polymer with peptides for targeting or dyes for imaging studies. So you know that I'm limited in the information I can reveal. So how do we achieve this objective? So first step is the synthesis characterization of the polymer. It's the first time I didn't have my excipients ready available for my manufacture. I have to synthesize and characterize the polymer using NMR to determine the degree of palmitoation and quaternization, GPC to know the molecular weight of the polymer, and HPLC to quantify the polymer in the formulations. Of course, all of you are familiar with the different techniques for characterization of formulations, DLS, zeta potential, TEM, or nanocytes. And of course, the most challenging one, evaluate the peptide proteolysis and formulation stability. For that, we use HPLC, size-excluding chromatography, and different other techniques to evaluate peptide proteolysis and formulation stability in freeze-dry or in suspension form. As you know, hydrophilic incorporation is a challenging process. I don't have time to get into the subject, but one small challenge is effectively and correctly quantify the drug loading, because you need to effectively separate the encapsulated drug from the non-encapsulated drug. An easy way to make sure that we don't have mixed fractions is to know how to quantify both the drug and the carrier. The drug is easy. You can use HPLC with UV detection. The carrier sometimes is difficult, maybe with some modifications of the polymer. But we optimize a method to quantify our polymer using HPLC by using an evaporative light scattering detector. Basically, the effluent from the column is nebulized and vaporized at the specific temperature that we selected, leaving the analyte in this particle form that can be detected by a DLS detector. Using this method, we were able to quantify both the polymer and the cryoprotectant once we have freeze-dry formulations. And currently, we are trying to optimize the method to also quantify the peptide in the same run. So what did I learn in these 8 months? No, I didn't learn how to make new friends. this is here just to emphasize one fact, because the quality and how much we learn depends on the people that we work. And I was quite lucky because, as you know, this project compiles most of top European labs in nano delivery research. And of course, my colleagues, amazing group, as one of my colleagues, Gang's always saying, is amazing. They teach me a lot, I learn a lot. And it allows me to discover new skills and learn new techniques, how to prepare a polymer that is quite different from using phospholipids available in a bottle, how to characterize, how to prepare polymer nanoparticles, because I was basically a lipid guy. Several new techniques that I developed, the new HPLC detector, nanosite, TEM. Probably most of you know how tricky it is to do particle stability in gastric and intestinal conditions. So basically, I'm adding one more step in my marathon for nano delivery systems. In terms of scientific output, since I began 8 months ago, the main focus one was delivery deadlines. But we have several ongoing experiments, probably ending this month or the beginning of the next. And depending on the results, probably a research paper will be issued. Also, of course, a review article presenting all these challenges of oral delivery of peptides is also a possibility. As to my next step, I'll continue at UCL under supervision of Professor Andreas Schatzlein and Professor Idioma, and my colleagues, that without her help it was not possible to complete in 8 months all the loose ends that I inherit from my predecessor. Thank you very much.
Okay, any questions? Frederick.
Thank you for this presentation. What are you using this evaporative light scattering detector for the HPLC? What is the aim of using this detector?
To quantify the polymer. Basically, it's to quantify the polymer.
Okay, but you cannot quantify the polymer with regular another kind of detector, regular detector, such as diode array detector, or what?
Yeah, but we can use refractive index, but this method is perfect because, for example, with this method, we can use gradients that in the refractive index is more difficult to use because the machine detects the gradient variations. So this method has this advantage. And also is not affected by the properties of the polymer. So if I change the polymer, it's possible for this machine to detect differences in the polymer. And as you can see, of course, there's a question of availability. This is the method that the machine that I have available in the lab, so it's also an issue. Of course, there are other possibilities, of course.
I was just wondering why. Thank you.
Silky.
No. No, so far the results are being reproducible. The only issue, but it's getting fixed, is that when you do a scale up, but so far it's being taken care of and is solved. So I didn't see in all the formulations that I prepare any significant variability. Of course, when you do scale up, as the first, I forgot about the name, the first student that presented, so that sometimes when you do scale up or scale down, we have some variations. We have in all the systems, but it's easy, as he presented, it is easily fixed.
No, misunderstanding. I'm talking about the toxicity of this positive polycarbonate.
The IC50 of this polymer is around 1 to 2 mg per mil. That I don't know the exact data for other positive polymers, but I work with positive phospholipids, and they were significantly more toxic than this. So compared with the other options that I know, I don't, as I saw, I'm basically a lipid guy until now. Compared with other positive phospholipids, for example, is relatively better.
Hi, up here, thank you.
Wait, sorry.
Yeah, so are you, as I understand it, you're making a self-assembled, like a complex class of it? of the chitosan with the negatively charged peptide?
Yeah.
So these are very sensitive to polymer properties.
Yeah.
So can you synthesize that the same way every time?
Yeah.
And when you make batch to batch, you make the same nanoparticles, or what do you think the prospects are for a pharmaceutical product in the future?
It's like this. We synthesize our batches in the lab. We confirm every synthesis. We confirm by NMR, the P and the Q, the palmitoylation quantization. So it's highly reproducible. We have SOP also for synthesis. We have specific amounts to ensure that we have specific palmitoylation quantization. There was, I don't recall the colleague that did the study, but we have specific like curves telling us that using this amount, this time we'll have this. So to ensure higher reproducibility. Of course, in the future, this project, it's possible to synthesize significant amounts of polymer. But I think these issues also arrive when people start using or describing liposomes, the issue of the reproducibility of the raw materials. And I think with the need, when the need appears, new improvements on technology will accompany that to ensure that we have polymers reproducible and then don't have variations in batches. But from my experience so far, I have obtained batches with with similar palmitoylation and quantization levels of substitution, and the particles are quite reliable in terms of properties. Even in terms of proteolysis stability, the results are the same from batch to batch, and from batch of GCPQ to batch of GCPQ. So far, I haven't found any problems.
Okay, thank you. Move on.